Sir, we are live now.
All right. Very good morning, everyone. Welcome to all the students of MOOC who are joining today. I'm Sanjeeva Srivastava, your instructor for the course. So as you know, the proteomics field is very exciting. We are trying to learn new tools and new technologies, challenging proteomics workflows every day. And as you're learning in this course of proteomics or proteogenomics, uh, how challenging it is to understand and unravel the complex proteome. There are many techniques which are trying to, first resolve these complex proteome mixture, and then trying to understand their function, their localization, how they interact, and various type of unknown function, and how to understand biology of it. So therefore, the proteomics is still limited with kind of technology which we have, but we're still trying to make best use of the available technologies. And in the course so far, you have been studying about sample preparation and gel-based proteomics, which is quite fundamental to the proteomics. But then as you move along, you have to see how best you can do much more high throughput proteomics. And that's where mass spectrometry-based proteomics and some of the functional proteomic tools will be important. So in today's live session, what we thought, we will first start with the very basic skills in the lab. In the MOOC sessions, we will try to give you some new activities or new things from the lab, which we are not able to cover in the course. So we don't want to make it another theory class here, but rather we will try to give you much more, overview directly from the lab that how you can do some experiments. Today, what we have planned, we are going to show you the workflow for doing proteomics, starting from the sample preparation. If you take any biological sample, how to prepare the lysate, how you can do different type of quality checks. You want to then quantify the protein. You want to do the standard SGS gel to look at their quality. And then if the everything looks good from quality and quantification, then you would like to look for the digestion of the protein. So there could be two approaches of doing that, either in gel digestion or in solution digestion. For the complex proteome mixture, it's actually better to do the in solution digestion, which we'll talk today. And after doing the digestion, you will get the tryptic peptides, which has still the salt and the buffer components, which you like to clean up. And then the desalting steps will be required. And again, after cleaning up the peptides, you still want to know that how much peptides you got, the yield. So you want to again quantify the peptide, and then you are going to do the mass spectrometry-based run. So in this manner, you will be uh, going through the workflow today from the lab. We'll also show you briefly about the gel-based proteomics workflow. Uh, Given that you have already studied quite a bit in the modules in the course, we will not talk a lot about it, but at least show you directly from the lab how SDS page setup and 2D gel setup looks like. But let me tell you, like how the field of proteomics is progressing is very rapidly, right? And newer and newer instruments are really bringing the unprecedented scale of proteome which we are now able to see. So from the same complex samples in maybe 2 hours run, a single shot injection can now give you maybe 10,000 protein, which is unprecedented, right? It was not thought of the time when I was doing PhD. So uh, I think, what you will see, like, in which way we can now try to be doing the smart way of proteomics when steps are minimal, so that our sample loss is minimal and our output or the number of proteome coverage is the maximum. So for doing so, there are three major things will be required. Number one will be sample prep. Number two will be instrumentation for data acquisition. And number three, data analysis. So the first part is always going to be challenging and important. How best you can get the good yield of your proteome without much losing your biological sample. And that's what we will emphasize today. But second part is something which is you will see the paradigm shift happening in the field, newer and newer instruments coming forward. Orbitrap technology revolutionized the field of proteomics. Now we have very recently, Thermo Fisher has launched Astral Orbitrap, which is probably one of the highest resolution with the maximum capabilities in the field. And likewise, Bruker has launched timsTOF Pro 2 setup, which is more capability of single cell proteomics, another revolutionary instrument. So again, like with these kind of capabilities and with a good sample prep, now you can achieve the very in-depth biology of your projects. And then, of course, comes the data analysis, which we are talking in the course, which will be also talking to you in much more detail in the next live session. So let's begin today's session. First, let me introduce now to the teaching assistants who are part of this course and who have been, running the course, who have been helping to take many of your queries. So we will have some short introduction clip for the TAs, and then we will move to the different sample prep steps from the lab. All right, let's first start with Ankit. So Ankit is a PhD student in the lab. He first joined actually as an MTech student, then continued for his PhD. He brings a lot of computational skills with him, and he's focusing on understanding meningioma disease biology by employing different type of LC-MS based proteomics and metabolomics tools. Uh, and currently he's trying to see like how best we can see like the multiomics analysis for understanding the meningioma better.
Hello, my name is Ankit. I am a third year MTech PhD dual degree student at IIT Bombay working in Proteomics lab under the guidance of Professor Sanjeeva Srivastava. My research topic is on one kind of brain tumor, meningioma, to identify the different altered proteome in the disease condition. So I am a PMRF fellow and is taking the live session on Introduction to Proteogenomics course every Saturday from 3 p.m. to 4 p.m.
All right, and next year is Deep Biswas. He's essentially handling the proteogenomics course, and Deep is almost towards finishing his PhD now. Uh, he has been contributing quite actively on the computational side in the lab. In addition to his work on meningioma, he's also contributed actively onto the brain prot, which was one of the very bigger initiative of making the brain prot database, uh, understanding various type of brain diseases. It's a good idea for you to check out brain prot, brainprot.org, and you will see like how best one could understand different type of omics information from this portal. So Deep has been looking into various type of computational workflows to understand meningioma tumor biology.
My name is Deepturu Biswas. I am currently the Senior Research Fellow in the Proteomics Lab of IIT Bombay. I am doing my PhD under the guidance of Professor Sanjeeva Srivastava. My PhD topic integrates the development of big data analysis pipeline and integrated omics analysis to understand the pathobiology of high-grade meningioma. In the course of time, I gained experience in understanding and developing big data analysis pipeline, machine learning concepts, strategies, concepts of proteogenomics, and statistical analysis. I hope you are enjoying and learning from the NPTEL MOOC courses of Proteogenomics 2023. Thank you.
All right, thank you, Deep. So next is Shashwati. Um, Shashwati is currently working on breast cancer proteomics, and especially in collaboration from her co-guide from Tata Memorial Hospital, Dr. Sudeep Gupta. She's trying to look at triple negative breast cancer and identifying the potential proteomic marker and the therapeutic strategies in Indian women. So her work again involves very in-depth proteome analysis, and she's one of the primary TAs for the proteomics course who have been interacting with you on Telegram and various other forum. Uh, and of course, she handles these proteomics tools and technologies firsthand in her own research. And she's also quite experienced TA for proteomics course, which I teach at IIT Bombay. So it's good to have Shashwati with you for this course, and I'm sure you are enjoying, her mentorship and various discussion along with her.
Good morning, everyone. Myself, Shashwati Pariyari. I am currently in fourth year of PhD in Biosciences and Bioengineering Department at IIT Bombay. I am currently working under the guidance of Professor Sanjeeva Srivastava. My PhD topic mainly revolves around multiomics characterization of triple negative breast cancer towards identifying novel therapeutic targets in Indian population. So I will be your TA for MOOC course Introduction to Proteomics. I am happy to take any queries from your side. So let's keep learning. Welcome to the MOOC course. Thank you everyone.
All right, and next year is Medha Gayatri Pai. Medha is working on medulloblastoma, one of the very deadly brain tumors, which has been less studied. It is actually one of the pediatric brain tumors, and Medha has been investigating both utilizing proteomics and metabolomic analysis of medulloblastoma tumors. She is quite expert of broad field of proteomics, but also very specialized into targeted proteomics, which is something which you will be learning toward the latter part of the course, how to validate the specific protein. So on one hand, with the global proteomics from the gels or mass spec, you want to identify the targets, but how to validate the target is another thing which you would like to learn. And that's where method expertise of single reaction monitoring or multiple reaction monitoring based assays from targeted proteomics are quite important, and she will be talking to you in that area. Medha is also quite experienced of both teaching my proteomics courses at IIT Bombay, and several times she had done the MOOC course. So I'm sure you are leveraging from her experience and enjoying discussion with her on this course.
Hello learners, welcome to the course. I'm Medha Gayatri. I'm a third year PhD student here in IIT Bombay in Professor Sanjeeva's lab. My work revolves around a pediatric brain tumor called medulloblastoma. I aim at utilizing high resolution proteomic techniques to understand the disease better. I'm also working on developing a targeted proteomic assay for better diagnostics of the disease. And we finally aim at clinical translation of all the knowledge that we are getting from this.
All right. Additionally, we have Shruti Ahuja as one of the PMRF candidate who's also doing the TA-ship for the course. Uh, she works under Professor Kiran Konda-Bagal's lab at BSB along with Siddharth Talur from Electrical Engineering. Her work is on developing a field deployable device for wastewater surveillance of infectious diseases. She is keenly interested to develop some of the proteomic assays, and I'm sure in the course she is both learning and trying to utilize her learning to further convey those details to all of you. So welcome Shruti in this course as a TA, and I hope all the participants are enjoying learning with you.
Hello learners, I'm Shruti Ahuja, a third year PhD student here at IIT Bombay in the CRNTS department. My research revolves around wastewater surveillance for the detection of infectious pathogens. Apart from that, I'm keenly interested in clinical proteomics and the use of proteomics for diagnostics.
All right, so you have already seen the course, of course, so we don't want to again spend a lot of time on it. But I hope you are finding the course is going with the quite decent pace. You don't have to rush too much. And from the week one, while you would have learned about the basics of again amino acids and proteins, to moving to the protein folding, protein misfolding causing different diseases, and then the week two was primarily involved into the sample preparation, which now you'll again see in the lab directly how that can be performed in the lab settings. And then we had moved on to some concepts of 2D technology, two-dimensional electrophoresis, followed by moving towards more of the advanced fluorescent-based techniques like difference in electrophoresis, DIGE. So this is the workflow, but I think some of these we would like to show you today in the lab directly. So without further taking any more time on this particular discussions, I think let's take you to the lab directly now and start discussion about how best we can do proteomics in the lab. Yeah, so the very first thing is what we will try. We will try to show you briefly the gel-based proteomics setup. Uh, Although, like, rest of the time we will try to mainly focus on your sample preparation strategies. But to begin with, we just want to show you how 2D gel setup looks like in the lab. Uh, so, as you have been studying in the your theoretical part of the course, that for separating the complex proteome with the gel-based proteomics, first you need to rehydrate your protein on the IPG strips, right? Immobilized pH-gradient strips. And what you see in the main frame in the lab is essentially one of the unit for doing the isoelectric focusing. There are different commercial makes of it, but this particular unit is essentially able to prevent the light. So it is, able to provide you light-sensitive environment, which is quite important for not 2D but mainly for DIGE experiments when you want to do fluorescent-based stuff. So this setup is required for the first dimension separation of the isoelectric focusing. Can we see the setup for the tray, please? So, of course, this is the bed of the IEF, but if we have the tray of the isoelectric focusing unit, that's where we keep the our IPG strips. And the very first thing which you have seen, like, you'd like to take out these IPG strips, immobilized pH gradient strip, which comes with different type of length. Sometimes it is pH 3 to 10, sometimes it can be 4 to 7. Depending on the your questions, you'd like to choose this kind of strip which you see right on the frame. And then this, you take a tray, rehydration tray, and in that tray you can put the protein, and then on top of that you can place the IPG strip. So that process can be done as a rehydration strip or the passive rehydration strip, which you will do overnight. And after you have done the rehydration of the IPG strip, then you will place this in unit, which is what you see here, the isoelectric focusing unit. And it is actually, what you see from the transparent thing there. Uh, Here from the outside, you can set up the program, you can set up the voltage, and you want to ensure that sufficient voltage hour is being attained when your IF is going on, right? So again, that requires some optimization. Depending on the how much protein you have loaded on this strip, what is the length of the IPG strip, you can go, depending on that, you can set up the volt hours. And most of the time for the large strip with 1 milligram protein, it will take maybe overnight time for IEF to get completed. So once the isoelectric focusing is completed, then you have to perform the equilibration steps, right? And that's where you have to perform equilibration 1 and 2. If you recall from the theory, you have to just move from one tray to other tray, these IPG strips. So please show the IPG strip once again. And so let's say after your IEF is done, then you will take this IPG strip and you will put in the equilibration tray, which contains the DTT or the, for reduction. And then you wash it again further with the second equilibration, which will be iodohistamide to prevent the any more reoxidation. After these two equilibration steps are done, then now you can place this particular IPG strip into the second dimension, which will be SDS page. Do we have a tan alt setup over there? So we can show second dimension separation setup. So what we see right now is a small unit, which can also be utilized, but this is a LGS page, a typical LGS page setup, where if you have these kind of comb, you can use the first dimension separation. If you want to use a flat comb, then you can put the IPG strip on top of that, and that's how this kind of thing can come. But we also have additionally very large running units, which is known as the standard 6 unit, where you can run 6 large 2D gels at the same time. And this kind of large IPG strip can be placed over there. But irrespective, the concept remains same. You can place your IPG strip on the SGSPH unit, and then you can separate it based on the molecular weight. Once you have completed the second dimension separation, then you can take out the transparent gel, and you are going to now further stain them with the Kumasi or silver or any kind of staining solution. If you were to do the dye experiment, where you would have added the dyes Psi3 or Psi5 dyes in the beginning, then after running your second dimension, you will directly scan the gel with the fluorescent scanner, and you will generate multiple image of Psi2, Psi3, Psi5 from those gels. So that is the dye concept. So just again, I think we wanted to refresh you and show you the setup directly from the lab. But today mainly we are going to focus on the sample preparation part. And now I request Shashwati and Medha to take over and start from the sample preparation steps of how to take biological sample and prepare the lysate and then take it from there. So Shashwati, over to you.
Good morning, everyone. So I'm going to show how to really prepare sample. So for this experiment, I have taken cell pellets. So you can use any kind of tissue or like plasma or extraction of protein. So for now, I'm showing how we are doing from the cell pellet. So I have taken the cell pellet here. So size the cells, we are using urea lysis buffer. What it will like lyse the cell and the protein which are inside the cell, they will come out and so this is a very easy. So I just add the lysis buffer. Here I'm adding urea lysis buffer. Then gently mix the cells so that it will mix in the buffer. Yeah. So you need the homogenized solution. After adding urea lysis buffer, one component that we are adding is PIC. So what is PIC? It is a protease inhibitor cocktail. So PIC, it will inhibit the proteases so that it won't affect our protein and our protein will be safe. So I'm adding the PIC in 1:10 ratio. If I'm adding like 200 of buffer, so then I'm adding 20 ml of the PIC. Then what we are using for the lysis here, sonication method. So here this is the sonicator. So I have set up the sonicator for 1 minute and the amplitude is 40 amplitude 40%. And the pulse is 5 second on and 5 second off. Because it will do the continuous pulse, then the protein can be degraded. So this step will be done on ice. So you can use a mini cooler or you can place your sample in ice. Here I am using a mini cooler to sonicate my sample. So the probe needs to be dipped into the solution. Then I'll start. So it will take 1 minute to sonicate my sample. So due to sonication, the lysis will happen and the protein will come out of the cell. It will take around 1 minute to the sonication. Then after sonication, what we can do, I stop it.
Now you can show that this solution is clear enough, and there is like no turbidence. So now how you can extract protein from this after sonication? What we does generally, we centrifuge the sample in 8000 rpm at like 4 degree for 10 to 15 minutes. Then after that, whatever debris will be there, it will be get settled down in the like bottom. So we will collect the clear supernatant from the sample. So those are our proteins. So you can store your protein at -80 for longer time if you want to use it like for further experiment, or you can carry your experiment. So it's required that your protein should be like kept at -80, so otherwise the protein can be degraded. So this all the process you have to do quickly, fast, because otherwise protein will cause problem. So thank you. Then we can show you in the Bradford where how we can
Hello, so good morning everyone. So as you have seen that after the sample is live, now we have got the, for example, in this case, so we have got the proteins extracted from the cells. Now we want to quantify or measure that how much protein is there in the samples. So for this, what we do is that we use the Bradford method, where we use the Bradford reagent and try to measure the fluorescence change. So for this, first we take the BSA, standard BSA, and we use different concentrations of the known BSA, that is the standards, with 0, 2, 4, 6, 8, 10, up to however you are trying to know that quantity, so that the range unknown protein samples range comes within that range. So after that, we take the BSA samples and then add 1 ml of the Bradford reagent here. That makes us our samples with the known concentrations, and we also add 2 microliter of the unknown samples and add the same PSA. And then what we do is that we take this in kind of 96 well plate. So you can see that we place it. So firstly we put blank where it is no protein, and just Bradford, then 2, 4. In this way we make the prepare the at the samples, and then what we do is that we take the readings in 595 nanometer in a multi UV with spectrophotometer, and then we prepare a standard curve and then try to find out the unknown concentration of the samples like this. So you can see that here is your amount of BSA that we know, and what is the average absorbance we have got, and the net OD, and for the unknown samples which we had added, so it is giving at 0.14. So from here, this curve with the slope you get, and you will calculate your unknown values for the x. And after and also the thing is to measure is that what we are doing the standard curve, we should have a good R square, so that it will show that our the experiment is fine, and then we can easily calculate the accurate estimate the proteins. The accurate estimation is quite important, because the next step that we would do for the digestion, so we need to know that how much protein we have done, and this step is just like very much crucial.
uh, so hello, Medha here. So now that we have done the quantification of the lysate, like the protein sample that we have prepared. So as Ankit has explained, we did it using the Bradford assay. So now that is ready. Now we need to do one more step before we proceed for the digestion of the samples. So first we have to run it on a gel and see how it is coming. So it's a standard SDS page gel. So page means polyacrylamide gel electrophoresis. So we have two, like we have different kind of plates like this. This is called a thick or spacer plate, and this is called a thin plate or short plate. So you stack them like this here, and it's put into this clip. Uh, yeah, so it's put it like this, and then you stack it on this casting unit. this unit is called the casting unit, and you clip this thing here. So now you can fill your solutions. Um, we have stacking, and we have different stacking and resolving solutions. So these are made using tris buffer. So we have different concentrations of tris buffer at different pHs. So for the resolving gel, that is the one at the bottom, like the one which resolves the proteins, that is at pH 8.8. And it is usually, depending on your application, you can make it from anywhere between 8 to 12%. And for the stacking gel, we need first, when we load the samples, we need that the sample be, all the bands in the sample be stacked together, and then they start resolving. So the top portion of the gel would be a stacking gel, which is at pH 6.8. So these are used to prepare the two gels, and other components are common between the gels. So that is like, one component is the 30% acrylamide or bisacrylamide solution, and there is SDS, obviously, because it's a SDS based gel. And then we have the other gelling components like ammonium persulfate and temate, which help in the polymerization of the gel. So these are the basic units, and that when we add it to the this thing, it helps in the polymerization of the gel. So we add our solution in this one by one, and we properly so I can show you. Once it is added, it will be filled till up, and then we properly place the comb. So in this way, we'll get grooves or wells in our gel according to the comb. So I can show it to you here. This is a prepared gel. So you can see it is an already prepared gel, and we have kept it ready for running. And here we have the wells, uh, which we'll use to load the samples. So once we load, now that we know what the quantity is, so we'll load around 10 to 20 μg of sample onto the gel, and then we'll check how the sample is looking like how good its quality is. So for that, after loading, I can show one loading also. So this is our sample, and we have added it with the dye. We have prepared it with the dye, and this is like between the two plates in the sandwich, you have to go and add the sample, and it will, because of the loading dye, it will go and sit at the bottom, and you can see that it is getting loaded into the gel. You can see that it is getting loaded onto the gel. So similarly, we load all other samples, whatever we want to analyze on the gel, and then we stain the gel overnight and de-stain it to see an image like this. So we have a gel like this the next day. I think it will be. So this is a gel that we have, but we'll show it to you on the screen. So here it is better visible on the screen. You can see that there is, as I explained, there is the running gel, which is at a higher pH, and also at a higher percentage of acrylamide. And then there is the stacking gel, which is used to stack the samples, like all the samples stack into one band. And then when they start running through the running gel or the resolving the gel, the bands will get separated. So this is how it works. And the SDS that we are using in the sample loading buffer and in the gel actually helps to give a uniform charge to all the proteins, so that they can travel only based on their size rather than their positivity or negativity of the sample. So this is how, like, it's a basic SDS page concept. Um, just showing you one of the example gels that we usually get in our lab. So if, for example, here you can see we have loaded different types of cell pellet lysates. So these are all different kinds of cells from different cancer cells. And you can see, if there is one cell line which is giving it's called U87, and it is, all these are loaded equal, like 5 μg, but you can see that the banding pattern in one sample will be different from the other. So this is the first step of analysis where you are seeing differences between your samples. You're able to visibly see that how your samples are differing. So for example, this particular very strong band here, it's very abundant in this sample, but here in this sample, it's not at all that visible. Again, in this sample, it is abundant. So similarly, we can check for if someone is interested in particularly this molecular weight of sample, they can then cut this band, do perform digestion, and then check for whether what protein is coming at that particular molecular weight. But today we are going with a cumulative, like we are not going to do selection of which band we are doing. We'll be taking the whole lysate and performing in solution digestion. So this is also a concept that you learned recently. So we'll be demonstrating that, like Shashwati will be demonstrating. Yeah. So now you got your protein. Now you QC check like the lysate is proper and there is no problem with your lysate preparation. So you can go ahead with the protein digestion, which will make the protein into smaller peptides. So for that, we are doing proteolytic digestion with the help of trypsin. So what is the steps involved in the digestion? So first is the addition of TCP or DTP that you can use to reduce the bond. What it will do, it will cleave all the double bonds which are present in the protein. So what we are, so first you need to prepare the sample. For that, you did the quantification. Now you can take 20 μg, 30 μg or 50 μg, whatever is your requirement, that much of lysate. Then you have to make up the volume with the help of buffer B. Then after that, you can use TCP as I have already mentioned. So after adding TCP to the sample, what we usually do, we keep it in the thermo shaker. So it is a dry bath with a shaking. So the temperature should be 37 and the shaking should be around 500 to 600 RPM. So the temperature is very important here. So with the TCP, we are keeping the sample for 1 hour. So after 1 hour of incubation, what we are adding, that is the IA. So IA does the alkylation. So it will prevent the reformation of those disulfide bonds. So after adding IA, what we do, we keep it in the darker area in room temperature for 15 to 20 minutes for the alkylation steps to happen. So IA is a light sensitive. That's why we prepare it in a dark tube and the incubation is in the dark condition. So you should not expose your sample after adding IA. So after like 15 and 20 minutes of IA step, what you can do, the main important thing is to check the pH of the sample, because the sample will only the trypsin can digest at a certain pH. So with the help of buffer B, so buffer B is at pH 8. So we need to measure the pH of the sample before adding to the trypsin, because only trypsin can act on a pH 8. So after adjusting the pH, you can add the trypsin to the sample according to your like percentage. So usually what we do 1:30 ratio digestion. If you are taking 30 μg of protein, then you can add 1 μg of trypsin to your sample. So now I have already adjusted the pH. Then after adding the trypsin, again we have to incubate our sample for 16 hours, like overnight or 16 hours in 37 degrees centigrade in the shaking incubator again. So the temperature here is also important, because at 37 degrees the trypsin can only cut in the respective site of the protein, like lysine and arginine. So that is the whole trypsin digestion state. So now you got your respected peptide that you wish after the proteolytic enzyme digestion. So after that, as I have already shown in the previous step, like during the sample preparation, we are adding like different kinds of lysis buffer. So it contains salts like urea and all. So we don't want those salts to hinder our mass spec. So for that, we are doing desalting step so that to remove all the unnecessary salts which are present in our samples. So for that, we are doing desalting step after digestion. So here we are using commercial columns, which are like setback columns. So here you can see this white part is the C18 material, which can be used for the desalting. So what we are doing here, then we will pass our sample through this column. So our peptide will bind to the column and all the salts and the unnecessary material that we don't want, it will like dilute out and the peptide will bind to the column. Then after doing like following few steps, like first we are adding conditioning solution. So it's 90% methanol in 0.1% Fa. So what it will do, it will first activate the column because the column is dry and there is nothing. So we need to activate the column before passing our sample. So this is the conditioning solution. I'll show how we do one. Like what we can do. So I'll just add conditioning solution to the column. So after adding the solution, what you need to do, you need to apply a pressure. So the bubble should not come in the sample. Just. So for that, what we can do, we can cut the tip. Yeah. So the sample should pass a very slow rate, like 1 drop per 1 second. Otherwise, the bed will get disturbed and the air will pass through the column. So we should not do the like allow the air to pass through the column. Otherwise, it will get disturbed and our like desalting won't be proper. So the time is very important, like 1 drop per second. So this is how we do our desalting. So I'll show after desalting, like conditioning our column, we use the equilibration solution step. So what it will do, it will equilibrate our column with the solution where our sample is. Like our sample is in equilibration solution. That's why we need to do the equilibration steps before storing our actual sample. So after equilibration, we generally load our sample. Then I'll do the same process again, like passing the sample through that column. So now our all the peptide are bind to the column and all the salts they are like in the waste. So then I'll perform one washing step. If there is any kind of salt remaining in the column, it will go away. Then after that, what I'll do, I'll add elution solution that will allow all the peptide to like elute out from the column and I'll collect it in a new tube. So that's the basic concept of the desalting. So now Medha will explain how we are going to do the peptide quantification for the sample production. Yeah. So now that we have the peptides ready, we have our sample ready. So it's now ready to go. The last step is now we cannot just simply give the sample to the mass spec. We need to first quantify how much we are giving because there is a certain load that a column can take. So our instrument is an LCMS based setup. And every column, every liquid chromatographic column has a maximum load. And whenever we are doing quantitative studies, we need to know how much we injected the sample and how much of peaks we are getting, how much of proteins we are getting for so and so sample. So usually in our lab, we inject around 1 microgram of sample. So for that, first what we do, we have this sample that was digested and desalted. Yeah, this is it. So after doing the desalting, we collected it in this tube, and so you can either after desalting, it is in very large quantity. So the elution is done in a very large volume. That is, we what we do is we take these peptides which are eluted, and we use a vacuum dryer to dry the peptides. So now they are dried, and now we add a 0.1% Fa. So I will show you. So this is 0.1% Fa. It is formic acid, 0.1% formic acid in water. So water is definitely MS grade water, because now everything is going into the mass spec. So we need to be very sure, right from the digestion step to until the mass spec step, that everything is very good MS grade thing. So usually, depending on how much you have done digestion, how you are confident how much peptide you will get, we reconstituted it in some Fa water. So I am reconstituting it in around 20 microliter of Fa water. So I'll add this Fa water into the dried peptide, and I'll keep it on a vortex for around 5 to 10 minutes, and it will spin. So because of the vortex, the peptides will get reconstituted in the Fa water. So because of the lack of time, I'll just show you how we do after the reconstitution. We are not waiting for the reconstitution to happen. So just imagine that we have got this reconstituted for 5 to 10 minutes. So we'll spin, because it's a very low volume, we'll spin it so that everything comes to the bottom. And now what we need is, we'll just take 2 microliter. So 2 microliters of our sample, and we will place it on this spectrophotometer plate called mu drop. So this is useful when we are doing analysis of very low volumes of samples. So here in our case, it is important. So I am adding the sample like this. Similarly, I'll also add the Fa water as my blank, because in that is what we have reconstituted the sample. And before adding the samples, you should be sure that you have cleaned the plate. We have cleaned the plate earlier itself, just for the demo purpose. So now these are the sample, and now I will take you to the. So this is our spectrophotometer called Multiscan. Here we place the mu drop plate, and it goes in we control it using a software. And what we are doing here is we are checking the absorbance. We are first setting up the plate. The first is the blank, and the second is the B2 is our sample, unknown. So now we'll take the reading at, we'll take 2 readings absorbance. That is, one would be at 205 and another would be at 280. So the reading, both the readings are important. The reading at 205 tells us the quantity of how much peptides is there, and 280 will tell the amount of contaminants or salts. So we'll tell the software to take the reading. Start. So it will take the reading. You can see the readings here. It is just loading. Yes. So this is the blank reading, and it is, we have added blank subtraction. So it is already giving the average and subtracting the blank. And then this is what we are getting for our unknown sample. So we have got the plate. So we need to ensure we need to ensure that we clean the plate as soon as we are done with the analysis. So always make sure that you don't use any tissue to use this. It's like very scratch this thing. So we use good quality lint proof tissue. Anyway, so the readings that we have got, uh, actually we have taken the 205 and 280 readings, and using the Scopes law we get how much concentration is there. So there is one particular constant that we get, that is epsilon, which is based on this formula, where we are taking both the 280 and 205 values, and based on that formula we are seeing how good the quantity of the peptide is, quality of the peptide is, sorry. So it should be ideally between a range of 28 to 34. If it is anywhere lesser and or more than that, it will not be good for the health of the mass spectrometer. It means there might be more salts or something contaminants. So if it is between this range and based on the concentration quantity that we have got, we will prepare the samples. So I'll show you how we prepare the samples. So now if I know that my sample concentration, for example, here I am getting 2 μg per μl. So I will reconstitute it with SA water, and I will add it to the mass spectrometer. So I will add whatever amount of SA is needed to make it up in the sample, and then we'll take around 15 to 20 ml of the sample and add it to a MS vial. So an MS vial looks like this. So we have to very carefully add it without making a bubble in the vial. If you can see it, we have carefully added it, and now this vial is ready to be submitted to the mass spectrometer. So this much we are showing for today's live session. Next live session, we'll go ahead on how we'll take this sample to the mass spec and do its analysis, and how we'll understand how the proteome is changing. So proteomics further. So sir will explain to you guys.
All right. Thank you very much, Shashwati, Medha, and TS for showing the sample prep, different steps involved. So as the students, you have seen that, okay, um, what we are trying to show you the very basic workflow and very simple ways of doing proteomics. Sample prep could actually involve different ways of processing, but we showed you one of the ways. You can always even come up with the newer ways of sample preparation. Many times people try with the TC acetone precipitation or trizol-based protein extraction. And there's different type of molecules get enriched if you are trying different type of extraction method. There is not one single method which one could say that is the best way of doing the protein extract and preparing the lysate. So you can always, if you want to look for more coverage, you can always try more than one method as well. Even at the time when you are doing the digestion, you can use the trypsin, or you can also use the cocktail of other protease enzymes as well, so that it will improve the efficiency. Sometimes a cocktail of trypsin and lysine can be much more efficient. Again, these are different tips and different steps which can improve your proteome coverage. But where the maximum proteome coverage can change is after you have done this peptide cleanup. You got a peptide mix. Whether you want to now fractionate these peptides. So if your goal was to really look at deep proteome coverage, it means you are looking at, can I get 10,000 protein from a given sample if there are 10,000 protein in that sample. So you have multiple options now to take this peptide, and either you can go with the high pH fractionation kits to fractionate it, use different type of chromatography to fractionate your peptide. Sometimes people also use the same concept what we have talked in the 2D gel, this kind of IPG strip, and use that for the off-gel fractionator based on the isoelectric focusing principle. So a simple concept of that is if you are separating from 3 to 10 pH your entire complex peptides now, um, that you can put different cups so in the 3 to 10 pH strip, and you can now take out, after doing the EF, you can take out the peptides from the smaller regions of 3 to 3.5, 3.5 to 4, likewise, and now you got many fractions corrected based on their pH. So again, that is one way of doing the fractionation. But standard workflow involves, like, people use different type of chromatography to separate and use that in the online setup. It means you can directly, keep separating, and that fraction will come to the ionization for electrolysis ionization, followed by the mass spec analysis. So these are the different ways you can think creatively from the sample prep, or during the digestion, or even post digestion of the cleaner peptide, how well you can be creative to improve the coverage of your proteome from the sample. And many times from same sample, if you're just trying out one workflow, let's say you're getting 5,000 protein, but if you have tried multiple workflows, you might be able to improve your coverage to 5,000 to 7,000 or 8,000, right? So these are the different things to try out. Of course, instrument plays very important role as well. You're using which type of technology, which type of instrumentation, what resolution is there. All that also helps you to think about looking at deep proteome coverage. So the next exciting part will be the mass spectrometers, which we'll talk in the next live session, where we'll show you how to now take these peptides and inject into the liquid chromatography, followed by ionization and mass analysis. Different type of concepts involved over there, and then followed by the data analysis. These will be quite interesting for you to follow up in the next class. The field of proteomics is advancing very rapidly. Uh, what you have been taught on and following up so far is about the global proteomics. But then the next thing comes about the quantitative proteomics, how to compare your healthy versus disease conditions and look at how many proteins could be differentially things are on the surface of the cell. Can we think about looking at cell surface proteome, enrich the only the cell surface protein, which is mainly a lot of membrane proteins and ruggable targets? Can we also think about cross-linking those protein and look at the conformational changes? The field is growing to look into single cell proteomics. Can we now enrich our, especially in the tumor case, when the cancer is very heterogeneous, can we look at a specific population and try to separate that for single cell, enrich that, and then do proteomics on that? So there are many type of interesting developments happening in the field. I'm sure as we talk more and we will go further, we will be keep trying to expose you into the newer field of proteomics. I hope you're enjoying the course and you enjoyed the today's live session. If you have specific queries, please don't hesitate to contact me directly on my email ID and also on the course forum so that we can take the discussion forward. At least, some of you have shown a lot of enthusiasm in the course and many interesting queries we are getting, which we are trying to reply back to you. Some of you are also asking me the future possibilities of coming to our lab at IIT Bombay. A simple answer is your performance may guide you to come back to us. So if you are going to perform well and you will be topper in the course, we will try to give you some opportunities to come to our IIT Bombay lab and do some internship or workshop opportunity in future. Thank you very much, everyone. Enjoy the day, and we'll be meeting you again in the next live session. Thank you.